In this video, we are going to study the multiple alleles. Before that, the multiple alleles, we study the Mendelian principles. In Mendelian principles, the assumptions or laws of Mendel are postulated, and these laws are failed afterwards. One example, codominance and partial dominance or incomplete dominance are studied. You understand well that law of dominance is failed. Next concept is one gene pair is carrying the information of one character. This assumption is again failed, and that is the title of this chapter, this point, that is multiple alleles. Multiple means more than two. Two alleles are considered by Mendel, but in the many examples we see the more than two alleles possible for a given gene. Allows for a larger number of genetic and phenotypic possibilities. The multiple alleles has many specific features which can be studied in this chapter. The multiple alleles always belong to the same locus, and one allele is present at a locus at a time in a chromosome. Means multiple alleles are there, but one individual has only one pair of allele. One is maternal and one is paternal. Multiple alleles always control the same character of individual. They are all for carrying information or genetic inheritance of same character. The wild type allele is dominant over other alleles. Always the group of multiple alleles consist of one wild variety and other mutant varieties, as well as one is completely recessive variety. Out of that, wild variety is dominant over remaining ones. There is no crossing over in the multiple alleles because they are present on same loci. In the series of multiple alleles, wild type is always dominant. When two mutant types are crossed, wild form cannot be recovered. The cross between two mutant alleles will always produce a mutant phenotype. And this law of dominance, degree of dominance, may be a serial one. One is dominant than the other and recessive to wild. In this way, the multiple alleles have these features. We can define the multiple alleles as a series of forms of gene situated at the same locus of homologous chromosomes and affecting same character. This is all about the characters and definition of multiple alleles. There are other forms are also there. They have more than two alleles, but there is some different characters. One type is isoalleles. Isoalleles in which a similar, but on testing it proves to be a different one. For example, blood group A. Blood group A person have three slightly different types such as IA1, IA2, IA3, which are similar but found to be different after testing. These are called as isoalleles. Another one type is pseudoalleles. These genes are so closely linked can be separable only by rare crossing over. Such genes are called pseudoalleles. These multiple alleles are present in plants, animals, human beings, etc. Out of that, well-known example is ABO blood group system in man, coat color in rabbit, wing type in Drosophila, eye color in Drosophila. This list is very long. Some has the large number of multiple alleles, but that character, that phenotype is carried forward. Out of that, first example we see the ABO blood group system in human. In human being, blood types are there. 4 blood types are there. They are carried or the governed by the genes 3. 3 alleles are there. One allele is called as IA. I stands for agglutination, and A for A antigen or A antigen present on red blood cells. Second allele is IB. It has B antigen on its red blood cells. And small I, it has no any antigen. A or B antigens are absent in this third allele. These blood groups or blood types are shown in following table. Each blood type or blood group has specific genotypes and its own antibodies. Blood type A has antigen B, antibody B. Blood group B has antibody A, and blood group AB has no any antibody. Blood group O has both type of antibodies. Their genotypes are mentioned in left column. The blood group A has 2 types of genotypes, homozygous IA, heterozygous IA small I. B blood group has 2 genotypes as IB or IB small I. The AB blood group has genotype only one, that is IB, and O blood group also has only one genotype, that is small I. These are on basis of presence of antigen and antibodies. The antigens are present on wall of red blood cells. It is nothing but one protein present in cell wall of red blood cell. In this diagram you can observe blood type A has A antigen, which is shown in green spike like structure. Blood group B has antigen B, which is blue dot like structure. These colors are only for your sake of convenience, that is for your understanding. Blood group AB has both antigens, and blood group O has no any antigen on its wall of red blood cells. In this diagram you can observe the antigen and antibody presence. The blood group A has the on its RBCs antigen A and its plasma consists of antibody B. The blood group B or blood type B has opposite condition. B has B antigens on its cell wall and A antibodies in its plasma. In case of blood group AB or blood type AB, it has both type of antigens on the RBC and hence its plasma has no any antibodies. Then opposite to AB condition is present in type O. The RBCs of type O has no any antigen. Hence its plasma has both type of antibodies. Same can be understood with help of this diagram. Antigens which are present on cell wall, antibodies are present in plasma. Plasma is a fluid part around the blood cells. It can be understood with this diagram. Same conditions are mentioned in different way. RBCs, plasma and its blood type. These genotypes of blood type shows a codominance in some cases as well as complete dominance in some cases. For example, if there is a cross between blood type A and blood type O, this cross shows a complete dominance. Means this cross gives results according to Mendelian principles. But if cross is made between blood group A and blood group B, it produces a new variety, that is not A or B, but there is a blood group AB. Means it shows codominance. And in this way, the one character of multiple allele is proven with help of such a type of crosses. Human blood type is an example of both codominance and a trait with multiple alleles. AB blood group is universal acceptor and blood group O is universal donor. Next point is rhesus factor. The rhesus factor, also known as Rh factor, get its name from experiments conducted in 1937 by scientists Carl Landsteiner and Alexander S. Wiener. This experiment involved rabbits, and these rabbits are injected with a rhesus monkey's blood. The blood of rabbit produces antigen, and that antigen is called as Rh factor. This rhesus factor is an antigen, or more specifically it is a protein that exists on surface of red blood cells. If person has either two positive genes for Rh or one positive and one negative gene, they will test Rh positive. A person will be negative only if they have both negative genes. Means Rh positive has two types of genotypes, heterozygous as well as homozygous. But Rh negative has a genotype only recessive homozygous. The relevance of Rh factor and ABO blood typing is most important. It is very important in terms of babies. For example, if mother is Rh negative, she make antibodies against an Rh positive fetus if the baby gets a positive gene from its father and agglutination takes place. That problem created and it may be tested before the birth. The ABO and Rh genes are only two of many blood antigens that are present in human RBCs and must be matched up for successful blood transfusions. Hence, Rh factor is important in blood transfusion as well as Rh negative mothers, and in this way the multiple alleles may show a multifactorial traits, that is referring to control of expression of trait by several genes and as well as several number of genes there is environmental factors. Many multifactorial traits show continuous distribution. For example, human height. Human height usually variable between 120 cm to 200 cm. However, these genes cannot be fully expressed without all the necessary nutrients from a healthy diet. Means genes are also responsible as well as environmental factors are also responsible, and the traits which shows variety of stages. It is true for many phenotypic characters. For example, color of eye, color of body hairs. Such a many characters are there which shows multifactorial genes. In this way, the multiple alleles are present for governing the one and only one phenotypic character, and hence the concept of Mendel is again failed that one gene pair is for carrying a one phenotypic character.